Hello and welcome everyone to this last podcast on the cytoskeleton. In this podcast we're going to talk about actin filaments. Often we'll also hear these called microfilaments. Remember that the actin filament subunit is the actin protein. As we begin to think about actin and its role, I want to mention that actin turns out to be a very abundant protein. It's about 5% of any given cell, or I should say at least 5%. In some cells, like muscles, it's 20% of all proteins. They help cells do lots of different things. They help cells crawl. And we'll talk a little bit more about this later. But for now, you may have a cell like this, and then it begins to extend in one direction. This is because of the actin filaments in it, driving it in that direction. They're important in engulfing material by phagocytosis. So if there is a smaller molecule or a large molecule out here, a phagocytic cell, like an amoeba or a neutrophil, will extend around the molecule and engulf it. It's important in cell division. As you have a cell beginning to divide after mitosis, actin will form a contractile ring to help divide these two newly formed cells. They're important in forming specialized cell structures like microvilli. So you may have a cell here like in the intestines that, in an effort to increase the surface area, will extend actin filaments outward to push the plasma membrane out, as so. And they're very important in contraction. Now this contraction is important in, say for instance, moving this cell, but it's also important in a specialized sense with muscle contraction. But actin's role in contraction isn't just in muscle cells. Let's talk a little bit about actin structure. It forms a long filament structure of these individual actin proteins. But together, it forms the actin filament. Just like with microtubules, actin has a plus end and a minus end. Because the actin proteins have a polarity to them, meaning one end is different than the other, the filament also has a difference in polarity. They are a little smaller than the microtubules. They're only about 25 nanometers in diameter. So speaking of that, let's think about some comparisons of actin to microtubules, MT for short. Actin is thinner, it's more flexible, it's shorter. Remember, microtubules can run the length of a neuron, so it can be upwards of a meter or longer. Actin don't form quite as long of a structure, though there's more actin in the cell, so if you were to take them end to end, it would still be, in that sense, it would be longer. But each individual one is shorter. So I should mention that last point just in brief here. Actin is more abundant. Now with microtubules, we saw that GTP was used in binding the monomers. With actin is bound to ATP. And similarly, as we saw with microtubules, this plus end will have more actin ATP. And then as you move down here, that ATP is hydrolyzed, and you'll have more actin ADP. Like we saw with microtubules, actin is more likely to be added to the plus end. So growth is preferred at the plus end. And you can still add to the minus end, but it's preferred at the positive end. This actin filament will begin to fall apart at the minus end in a similar way that we might see it with microtubules. And that is because when ADP is bound to actin, there is a reduced affinity for other actin proteins. So I want to draw a cell down here, and I'll draw a couple actin filaments, and we'll see them crossing in various directions in the cell like I'm showing here. But we'll also see lots of actin proteins here that aren't in this filament. In fact, actin subunit are always at the critical concentration to allow growth. If that's so, the question is, how is it that actin doesn't always grow? We always have an abundance of these polymers at a concentration that it should grow and extend to the positive end with ease. How do we prevent that from happening? There are two strategies. The first is using these profilin proteins and thymosin proteins. These bind to the monomers, and as long as they're bound to the monomers cannot bind to the filaments and extend the length. But they're always present so that this actin filament can grow quickly when needed to, just by removing the profilin or the thymosin. Another way this is accomplished is by using capping proteins that will bind to the plus end of these filaments, preventing addition of monomers. So to describe some of the other features of the actin filament, how it grows and how it can extend plasma membranes, I want to talk about the general phenomenon of cellular crawling. Lots of cells crawl, lots of our cells crawl. I'm going to give three quick examples. The first are neurons during development. As neuroprogenitor cells begin to form neurons, they have to extend a long distance to form that mature neuron. Next would be amoebas. These are carnivorous, free-living cells that capture food by crawling along a surface. Neutrophils within our own immune system will exhibit cellular crawling. In all of these cases, you'll have a cell that is attracted to some sort of chemical in the extracellular space. And these cells will then move towards this chemical signal. And they will typically do so by extending part of their plasma membrane in that direction and then ultimately moving the rest of the body of the cell with it. Cellular crawling has three basic steps. Cell extends its plasma membrane towards a chemical, some kind of signal. This cellular protrusion that occurred in step 1 now adheres to the surface. Then the third step is the rest of the cell moves forward. It does so by releasing the adherence to the surface, but also contractile actin in it will help move it forward. We'll see that all of these events here are all dependent upon actin. Now before we begin, I want to show a quick video that demonstrates cellular crawling. What we see on the right half of the screen here is a human neutrophil. And over here we're going to see a pipette that's going to place some chemicals here that it's going to attract the neutrophil. And as it's released the chemicals, you'll see the neutrophil moving towards these chemicals. What we see are those three steps. We saw the protrusion of the cytoplasm moving forward, and then we see the whole rest of the body of the cell moving with it. Again, moves forward and the whole body of the cell moves with it. Pay attention to how it protrudes the cell membrane and how the rest of the body of the cell moves forward. And in this particular example, they're being a little cruel with these neutrophils and making them move in opposite directions. But you can see how quickly they can change directions based upon the chemical signaling. Okay, so let's look at this in more detail. And let's look at this first step of cells protruding the plasma membrane towards this signal. So you'll have your cell here, and you'll have various signals over here that it's going to move towards. What happens is that the actin filaments will move in this direction. However, the way it does it isn't just in these straight parallel lines like I'm showing here. Rather, it will do something more like this, because it's trying to move not just a very small part of it, try and move the whole cell. So you'll see actin filaments going in multiple directions and often connected with each other, so that it will slowly send the plasma membrane in this direction towards this chemical signal. This is called a lamellipodia. This almost sheet-like protrusion of the cell in a direction towards that signal. Now, alternatively, we can form a much thinner protrusion, like so. In this case, we don't see the actin going in these multiple directions, forming that kind of mesh work there. We will see them all going in the same direction, because you're making this nice, fine, thin tubule moving in this one direction. Or extension. I shouldn't call it a tubule. An extension moving in this one direction. And these are called, when they're thin like this, filopodia. These filopodia are very common when we're talking about the neuron development. Whereas the lamellipodia are seen often when we see that amoeba moving or the neutrophil moving. I want to zero in here on this lamellipodia here and looking at these actin, and I want to talk about some of the proteins that are involved. So I'm just going to draw part of the plasma membrane like so. And I'm going to draw an actin filament going this way. Now the cell wants to move in this direction, but this actin is going in the wrong way. So in an effort to extend the actin towards the plasma membrane, what happens is we have a nucleation site forming, and we'll just draw one here. So I'm going to write nucleation site. It forms because we have this protein called ARP2/3 binds here. And this is where the actin will extend from. But it can't do it on its own quite yet. It requires another protein. And that other protein is called WASP. It binds here, and it's necessary because it activates ARP2/3. And once ARP2/3 is activated, the actin can add the actin subunits to it and extend it in this direction. Now I didn't show it here, but we would still have those subunits all around here, the actin subunits, that would at this time also lose that profilin cap to it so that they can be added to the ends here. Once the actin filament has reached its destination and isn't going to push any further, we would finally have those capping proteins bind to it. So again, a nucleation site is established when ARP2/3 binds here. ARP2/3 is what's going to extend the actin filament, but it can't do it until WASP comes along here, the green component here. And once that activates ARP2/3, actin subunits will bind at the nucleation site and move in the direction of the plasma membrane. Okay, now let's talk about the next step. And remember, that next step was to anchor or adhere this lamellipodia. And the way it does that, I think it's going to be easier to show this cell from a slightly different angle. You can imagine that as we're looking at this cell, we're looking down on it. But let's look at it from the side view. So let's look at it from this view right here that I circled. And so we'll draw our plasma membrane, and here is the surface the cell is moving on. Earlier in this semester, we talked about these proteins called integrins. And integrins are transmembrane proteins. And they're an important component of that cell cortex. And what these integrins are going to do is they're going to provide a mechanism where this protruding actin will interact with these integrins. Let me write integrins over here so we don't forget what they are. They interact with them in such a way that they push them and allow them to interact with the surface. In doing so, it produces a nice anchoring mechanism where the integrins associate with the surface and the actin cytoskeleton. So what this effectively does is it allows this protruding part of the cell to anchor itself to the surface. And that allows this third step to come along. And that third step, remember, is to release the anchor of the cell body. So as this part's being held to the surface, this other part here, which at one time was attached to the surface, will now release itself. And as it releases itself, it allows it to kind of catch up with the rest of the cell. So it binds, it releases, and now as we're moving forward here, the whole cell will catch up with this part of it. It'll still be attached to the surface as we see down here, but the rest of the cell catches up with it. And then it repeats itself. It'll release these attachment sites. These attachment sites down here will form. These will be released, and now the cell can once again protrude towards these sites. It attaches, these are released, and it moves in this direction. Now the way I just described that, with these releasing down here, and it just kind of flows forward, part of that's correct. But the part that is a little bit more complicated that I haven't talked about yet is that we have actin filaments in here that are going to contract, that helps facilitate the movement of the cell in a more precise way. So let's talk a little bit more about how these actin filaments can contract. Okay, so let's continue this discussion of how that cell crawls using these myosin motors, or these actin motors. So let's begin our discussion of actin filament contraction and myosin motors. These are the proteins that are going to bind to the actin filaments to allow for that movement. So if you remember when we talked about microtubules, we talked about kinesins and dyneins, and how kinesins moved in the positive direction and dyneins moved in the negative direction. Well, there's only one kind of motor protein for actin filaments, and they are this family called myosins. There's about 20 members in this family. Most, not all, but most move in the positive direction. Structurally, we could draw the myosin as such, where here we have the head domain, and here we have the tail domain. The head domain has actin binding abilities. This is the part that will bind to the actins, and it binds and hydrolyzes ATP. Similarly, as we saw with dyneins and kinesins, the tail domain binds cargo. Anything that might be attached and needs to move with the actin will bind to this tail domain. And so this is where we see our greatest degree of variability amongst the different kinds of myosins. As I mentioned, there are 20 members, but there really are two large subfamilies, myosin 1s and myosin 2s. Myosin 1s are found in all cell types and are responsible for that cellular movement, the crawling that we just described. They're also important in establishing and maintaining those microvilli that we see in the intestines. Myosin 2 are specialized, and they're only found in muscles. So now to think about how these motors work, I want to show a quick video here. So as I start this video, in green here are the myosins, in red are the actin. Attached to the actin is the head portion of the myosin, and the tail portion here is attached to what is here is just a glass slide. But this could attach to various kinds of cargo or to various structures in the cell. As we see here, ATP is added onto this cover slip, and ATP will bind myosin, and as it's hydrolyzed, it will change its conformation set in such a way that it moves the actin forward. And this is showing the actual data. And you can see the actin, I'm going to pause it here, you can see the actin kind of moving around this slide, almost like a snake cruising around here. And what's not shown here are the myosin motors that are attached to this slide, moving it in this direction. So now let's bring it back to the cell crawling. Remember, you had this cell that was moving in this direction here, and it was moving along this surface that it was attached to. And remember, we said we had those integrins attaching to the surface here and the cell, and down here they've released those attachment sites. And we said that the cell would then move forward. But then we brought in the contraction activity of this filament, like so, with the myosin attached to it. Remember that myosin is attached to it, I'll just draw it like this, pointing in a direction towards the left. As it hydrolyzes ATP, it's going to move in this direction. And as it moves in this direction, with it being attached to the actin, it's going to move the actin all in this way. And since this is attached, the motor is attached to the plasma membrane, and the cell is no longer attached to the surface, it's going to move the whole cell in this direction. And it's a little misleading because there's not just one here. There would be many of these myosins that would collectively all hydrolyze ATP and move the actin, contracting it in this direction. And I think to show that in a little bit better version than my drawing, I know my drawings are awesome, but I want to go to an animation that I think will show it a little bit better. So as you will see in this animation here, here's our myosin with the motor head here. It's going to bind to the actin with the tail here attached to the plasma membrane in this case. And as you'll see here, it binds to the actin. And as you'll see here, as it binds here, the ATP is hydrolyzed, leaving ADP behind. And you see this conformational change. ADP is lost, ATP comes in. It loses its grip on the actin. ATP is hydrolyzed, it kicks forward and binds to actin, releasing that phosphate group. ATP comes back in, changes conformation, kicks forward, and it's going to bind to the actin. When it binds here, it hydrolyzes ATP. ATP comes back in here, a new one does, and it keeps moving forward. And as it's doing that, you can see that it is sending that actin in that one direction, ultimately resulting in the cell moving along here. Okay, the last thing I'd like to talk about is something called actin severing. And this sounds just like what it is. You may have an actin filament growing this way. So you can have these proteins called gelsolin and cofilin that will bind to the actin and facilitate its breaking. So you end up with two filaments, like so. And it can break it down even more. So one of the purposes of doing this is for depolymerization. And the other role of cutting these actin filaments is to make new sites for actin growth or polymerization. So if it cuts them up at multiple places, you can imagine having this pool of smaller actin filaments. And if the capping proteins are removed, you can imagine how we could have new growth of these severed actin filaments. So it has a role in depolymerization and a role in creating new sites for new polymerization. Okay, that's all I have for this last podcast on the cytoskeleton. If you have any questions, please let me know. If not, I'll see you in class. Bye.